Welcome to the Compendium of Knowledge, a living library of ideas, values, and insights meant to spark curiosity, open our minds, and help us see the world with greater depth and humanity. For our very first conversation, we are joined by Dr. Richard J. Roberts, who won the 1993 Nobel Prize in Physiology and Medicine alongside Philip Sharp. His discovery completely changed how scientists understand genes. He found that our DNA isn't just one smooth, simple code. It's full of hidden breaks and rearrangements that reveal just how intelligent and complex life really is. Today, Dr. Roberts serves as the Chief Scientific Officer at New England Biolabs, leading groundbreaking research in molecular biology and genetics. Beyond the lab, he's been a powerful voice for using science to tackle global hunger, improve health, and inspire young minds to see discovery as something that belongs to everyone, not just scientists. Dr. Roberts, it is an honor to have you with us. Welcome to the Compendium of Knowledge.
Well, thank you for having me.
So, firstly, for a young audience, could you explain in simple terms what you discovered about genes that led to your Nobel Prize, and why that discovery changed the way we understand biology?
Well, the basic essence of the discovery was that in bacteria, DNA is copied directly into messenger RNA, and that messenger RNA is then transported to an apparatus called the ribosome, which reads the bases in the messenger RNA 3 at a time and puts an amino acid into a protein. And the ribosome then makes protein based on the instructions in the messenger RNA. Now, all of this was worked out and discovered in bacteria. And when we came to ask some rather simple questions about a eukaryotic virus, not a bacterium, a eukaryotic virus like us, then what we discovered was that the RNA was made in a very different way from the way it is made in bacteria. First of all, the DNA was copied into a very long piece of RNA. And that long piece of RNA, instead of going intact into the ribosome, was actually cut and spliced into pieces before it gave the RNA, which we call the messenger RNA, that goes to the ribosome. A good example of this that most people understand is that when filmmakers make a film, they take a few shots, and then a few more, and then a few more, and they mix and get all of this in the editing room and splice it together to make it look as though these individual shots were just continuous. And this is exactly how eukaryotes, higher organisms, how they make the messenger RNA that then codes for the proteins.
Wow, that's fascinating. Thank you for explaining it in a very simple manner. And in previous interviews, you have mentioned that your prize actually came up from a previous experimental failure. What has this experience taught you, and what was this experience firstly, and what did this experience teach you about life?
Well, I think the key thing is, and something that I always tell students in particular, and especially their parents, is that failure is often a very good thing. Failure is when we learn. Okay? If you never fail at anything, you don't learn as much as if you fail, figure out why you failed, and then go on and don't fail again, don't repeat the problem. And that was what happened with us. We were doing some experiments thinking that RNA made in adenovirus, in eukaryotic cells, was made in exactly the same way it was made in bacteria. And we were actually interested in some signals in the DNA that were immediately preceding where the messenger RNA was being made. It's a region called a promoter, where the enzyme that makes the RNA polymerase, binds to the DNA and then starts making RNA. And we just wanted to see if a promoter in adenovirus was the same as promoters in bacteria. And we were expecting it would be, and so we came up with what I thought at the time was a very clever experiment to determine where that start point was, but the experiment failed. It didn't work. And so it, what it meant was that either we'd done something wrong, and we quickly found out if we hadn't done anything wrong, but that nature was actually trying to tell us something. It was trying to tell us that the hypothesis we had, that RNA in higher organisms was made in exactly the same way as that in bacteria, just wasn't true. It was being made in some different way, some way that we then went on and figured out how it did it.
Understood. Thank you. And for the listeners out there, you are viewed as a role model and have had a very fulfilling life. But now I begin to wonder what originally motivated you to explore genetics and molecular biology, if not for a Nobel Prize?
Well, it never occurred to me to go after a Nobel Prize, and I always encourage people not to think that that is ever an ambition they should have, because almost every Nobel Prize winner will tell you they were not looking for the discovery that they made. It was luck. They were trying to find something, didn't work, they looked to see why it didn't work, and made a big discovery. Now, in my case, when I was a kid, I was interested in mathematics, loved mathematics, and then I read a book about how to become a detective, and I thought that was a lot of fun. It sounded as though this would be a good ambition. I thought I'd be a detective, and then my father bought me a chemistry set, and I discovered that I could do all sorts of interesting things with this chemistry set. This was in the days when the chemistry sets were actually quite interesting. The politicians and bureaucrats had not managed to take out all the dangerous chemicals. They were still there. And I discovered that I could make fireworks, I could make explosives with this chemistry set, and so I did. And I loved it. I fell in love with chemistry. Went to university for chemistry, and in my third year as a PhD, I had really got all the results I needed for my thesis, and I spent a lot of time in the library. And I discovered that there was a journal called the Journal of Molecular Biology that was describing the origins of molecular biology, and one book in particular was by John Kendrew called The Thread of Life. And I read this book, and it explained in some easy detail that anybody could understand how molecular biology came about as a subject. It was about the origins of molecular biology. And by the time I finished that book, I knew that was what I wanted to do with my life. And so when it came time to do a postdoc, I applied to a number of labs, half a dozen labs, where they were chemists who had sort of switched into doing molecular biology or doing biology, but from a chemical standpoint. I applied to 6 places, only one guy took me, and that, as a result, I ended up at Harvard and started doing molecular biology.
Wow, thank you. And you previously mentioned the importance of failure, but in your
Mm-hmm.
entire professional life, if you had to give one lesson, one thing you think would be most beneficial for the youth of today, what would that lesson be?
Well, I think one of the things that I'm a great believer in is luck. And I think if you or anybody goes back and looks back on your life and say, well, when did luck send me one way rather than another? And I've done that many times. Look back, and I've discovered that much of what happened to me was luck. But the secret of luck is not to feel guilty because you had it and your friends didn't, but to take advantage of it and to figure out how you can use it so that this will so that this will make your life better. That's what I do. That's what I like to do. And I learned that when I was about 16, 17. I was playing snooker. I was a very good snooker player, and I was the West of England junior champion actually. But I was in a tournament, and I was trying to pot a ball and it didn't work, but I potted another one by mistake. And the next shot, I didn't pay enough attention, and I didn't manage to continue the break. And this old gentleman came up after me afterwards, and he said, you know, If you had taken advantage of that lucky shot, which you do by concentrating twice as hard on the next shot, you would have done really well. And he said, you're a very good snooker player, but you've got to learn to take advantage of luck. And I learned that when I was 16, and it never left me. Whenever luck comes my way, I always try to make the most of it.
Wow. That's very unique advice and something that not many people hear these days.
Mm-hmm.
My personal impression,
It's really worth looking back. You should do it yourself and encourage all the students to do it. Look back and see, and sometimes you're not aware of it until you look back. When I was 2 years old, I was living in Derby, and this was during the Second World War, and I was on a direct route from Germany to the Rolls-Royce factory where they were making the engines for the planes that were fighting Germany. The Germans were trying to bomb it, and they kept trying, but they were just not that accurate in those days. And as a result, the street on which I lived, one side of it was almost completely destroyed, but I was on the side that was saved.
wow.
That's luck. That is really, really luck. But I was too young to appreciate it at the time. But looking back, I've had many, many examples where luck has played a huge part in my life.
Wow, thank you. And my personal impression is that Nobel laureates have always had one thread in common, which was to achieve something bigger than themselves. You have often spoken about using science to make the world better, from your discovery of split genes to your work helping people understand how science can improve food and health.
Mhm.
Now that you have achieved that recognition, what are you doing to empower or motivate the youth of today?
Well, by doing things like this and talking to them. I like to go to conferences where there are lots of students. I like to talk to students. And one of the things I like about students is they very often ask silly questions. But sometimes I don't mind. I'm more than happy to answer them. But sometimes those silly questions are things I don't know the answer to. And that's when you start to learn. When people ask you questions that you don't know the answer to, you can go and look and see and find out. And maybe you have to do some experiments. Maybe you have to do some research. But I love simple questions. I love being asked questions.
Understood. Thank you. And hearing your story is inspiring, Dr. Roberts, and now we would love to hear from you directly. As part of your learning module, could you take us through your discovery and help us understand what could be called the language of life? How your work on split genes revealed that our DNA can cut, rearrange, and refine itself, which reshapes how we understand biology.
I'll take you through what happened when we did the experiments that led to the discovery of splicing. And I call it the serendipitous discovery of splicing because we weren't looking for it. We were looking for something totally different. What had happened was that a postdoc, Richard Gelinas, his picture is here, started working with me on adenovirus 2 messenger RNA. And we were rather interested in thinking whether a promoter, a eukaryotic promoter, the sequence that is on DNA that tells the RNA polymerase where to start making the messenger RNA, we just wanted to know, was that the same as the one in bacteria? And there were two in bacteria that had recently been discovered, one by Wally Gilbert's lab and one by Mark Potashny's lab. And they were fairly similar. And we thought, well, maybe the adenovirus promoter is going to be the same. I should say adenovirus is a eukaryotic virus. It lives in human cells. And it's one that's rather nice to work with because almost everybody has been infected with adenovirus when they were a young child. Their mother thought they had a cold, they recovered, and then you're immune for the rest of your life. So they're really very nice to work with in the lab. So we started doing that. And what we noticed from what little was known about adenovirus 2 is that early after infection, when the virus first goes into the cell, it makes four major RNAs called E1, E2, E3, E4. And the adenovirus genome, the DNA, is a linear DNA. And so the E1 starts at the left-hand end of the DNA and is transcribed, the RNA is made by copying the DNA from the very end and moving in. In the case of E4, it is being made from the other end. And so what we thought is that if we could find the sequences that were present at the two ends of the messenger RNA, E1 and E4, then all we needed to do was to determine the DNA sequence at the two ends of adenovirus, and then we would know where the promoter was. And I had another postdoc, John Ahrens, who was actually sequencing the DNA at the ends. So we tried to do this experiment, and basically the experiment was a simple one, was to take the messenger RNA early after infection, of which there were 4, treat them with an enzyme called ribonuclease T1. And ribonuclease T1 cleaves after every G residue. You know, there are 4 bases in RNA, C, U, A, and G. And ribonuclease T1 just cuts after all the G residues. And so if you do this on a messenger RNA, you will get 3 kinds of products. You get one from the 5' end, which has a triphosphate on it. The ones from the middle don't have anything at the 5' end, and the ones from the very 3' end of the messenger RNA have poly A, because that is put on the messenger RNA after it's made. And so all we needed to do was to find the 5' triphosphate containing oligonucleotide. That would be the start of the RNA, and then we could get the sequence and see where that was on the DNA. However, we had problems. We couldn't isolate enough early messenger RNA to do the experiments. It turns out adenovirus just doesn't make a lot of early RNA, and it didn't make enough that our experiments could be done. And so then we turned to looking at the late RNAs. And the late RNAs are shown on this diagram as L1, L2, L3, L4, L5 at the top. And these are all made and are mapped at different positions in the genome. And they're all of different lengths, and their position is different. And so we thought, well, if we could get the 5' ends of these messenger RNAs, then we would have to do some sequencing from within adenovirus, but it was probably still doable. So it wasn't going to be as nice as the early ones, but it should be okay. And so we were expecting, when we did the ribonuclease T1 treatment, that we would get maybe 10 or 15 different 5' ends corresponding to the start of all of the messengers. So while we were thinking about doing that, a key discovery was made. And this was made by several groups, but notably Aaron Shatkin, who was a very good friend of mine. And what they discovered was that this triphosphate that we normally associate with being at the 5' ends of messenger RNAs actually was modified. And there was a guanosine residue that was stuck on it in an inverted position, but it had 2 hydroxyl groups, 2 OH groups sticking out of the ribose ring shown on the left of this picture. And because of that, that meant that there was actually a very easy way to isolate these 5' oligonucleotides. And so that's what we did. So here was the experiment this time, same experiment, but this time we're looking for the 5' end with 3 M7G stuck onto the triphosphate. And I came up with an equally clever way of isolating these, and we did the experiment expecting to see maybe 10, 12, 14 different terminal oligonucleotides. But that didn't happen. What happened instead was that on the left-hand side, you can see there are two spots, A and B, and on the right-hand side, just one spot. And this depended upon how long after infection you looked at it, and it turned out that the A and B were actually the identical oligonucleotide, except the two hydroxyl groups, one of which was phosphorylated, had cyclized. They were related to one another, but they weren't different sequences. And so what this said was that every single late messenger RNA, of which there were 10 to 15, had exactly the same 5' terminal oligonucleotide. And that was puzzling. And it was puzzling because we thought the experiment had failed. In fact, when Richard Gelinas first did this experiment, I made him repeat it. And he repeated it 3 or 4 times. And then I said, well, let me do it and I'll show you how to do it properly. And I got the same result. And at that point, we believed we got a result. There was something going on in adenovirus that was new. It was new to biology. It was new to us. And we wanted to know what it was. So we started to do a whole series of experiments. So we hybridized messenger RNA to different fragments of the adenovirus 2 genome, and they all gave exactly the same cap, even though they were individual and separate messenger RNAs. And then a collaborator, Dan Klessig, purified messenger RNAs for a couple of well-known proteins, and they had this same capped T1 oligonucleotide. But then something strange happened. If you hybridize the messenger RNA to the DNA that is coding for it, you expect to get a perfectly stable RNA-DNA hybrid. But if you treated that hybrid very gently with ribonuclease, then the cap structure was lost. And that said that this cap structure must be coded in a different region than the messenger RNA. So the messenger RNA that had picked up this cap, the cap wasn't coded right next to it. And then Saida Zain did some experiments with adeno 2 SV40 hybrid viruses, and she showed conclusively that the capped oligonucleotide wasn't coded next to the main body. And early RNAs have a different terminal oligonucleotide, but that's not lost by mild ribonuclease treatment. And so that was a little strange too. So the question was, how can we explain this? Well, the explanation we came up with was that a typical messenger RNA in adenovirus, shown here at the top, if you hybridize it to the DNA that codes for it, then it should have 2 tails on it. One is the poly A, which we know is not coded on the genome, it's added after transcription has taken place, and a 5' end that must come from somewhere else. And we didn't know where it came from, we didn't know how it was possible that it got there, and we didn't know what it was. And so we devised an experiment, and the experiment is called an R-loop experiment, and is shown at the bottom. If you make a hybrid between RNA and double-stranded DNA, then it turns out that the DNA-RNA hybrid is more stable than the double-stranded DNA. And so you can form these funny loops called R-loops, and they're quite stable. And realizing this, we came up with something called an R-loop experiment. The RNA here is shown in blue. It's got the poly A at the 3' end, and at the 5' end it has this piece of RNA that contains the cap that is the 5' end of the messenger RNA. And we thought if we could find the piece of DNA that would code for this RNA, for this cap structure, then we could look at it in the electron microscope and show that this was in fact what was going on. And so I thought of this experiment one Saturday morning, and we decided that this looked as though it might tell us what was going on for the 5' end. And fortunately, although neither Richard nor I were electron microscopists, just down the hall from us were two very good electron microscopists, Louise Chow and Tom Broker. And we went down and we talked to them, described the experiment shown up here at the top, R loop, and then labeling with single-stranded adenovirus 2 DNA as a probe. And I had made a guess as to what I thought that might be. And then we said, if we make the reagents, could you do the experiment for us? And they looked and they said, well, it's not been done before, but yes, it should be doable. And so we made the reagents, and on the Tuesday morning, they did the experiment, or Louise Chow did the experiment. And what she found was that, sure, this probe DNA we had prepared did in fact hybridize to this 5' end of the messenger RNA, but it hybridized in two separate places. There was a loop in between. And this was the discovery of RNA splicing. That what was going on was that the RNA that made up a typical adenovirus messenger had at least three different origins in terms of where it was coded. The 5' end was coded at two separate positions on the DNA, and the main message was coded where it had been mapped previously. And this was a huge discovery. And every molecule that Louise looked at looked exactly like this. And so after that, she went on, excuse me, did quite a lot of work, and we eventually published this paper called An Amazing Sequence Arrangement at the 5' ends of adenovirus messenger RNA. And this was the discovery of RNA splicing, which was made in 1977. It was actually made in early March, but the paper didn't come out until September. And I have one little story here that I would like to tell you. When we wrote this, we wanted to say something descriptive about something strange and odd going on at the ends of adenovirus messenger RNA, and we looked at various adjectives that we could use, and I liked amazing. And the other authors didn't like it very much, but eventually I convinced them that we should use it. But then when we sent the paper out for review, the reviewers, I think there were 3 or 4 of them, all wrote back and said, hey, this is a great paper, fantastic finding, but you can't use the word amazing in the title of a paper. This isn't scientific. And so the editor, Ben Lewin, for Cell at the time, wrote and said, paper's good, just make a few changes that the reviewers had recommended, but you've got to change the title. You can't use amazing. And so I called up Ben Lewin, and we had a little discussion. I talked about some of the other things that the reviewers had mentioned. And then by chance I said, Ben, when you first saw this, what did you think? And he said, oh, it was amazing, he said. I said, I rest my case. And that's how we got amazing into the title of this paper. And as far as I've been able to discern, this is the first time that amazing has appeared in the title of a paper. I know there have been a few since then, but I've not managed to find one before. So that was kind of fun, and this was the paper that eventually led to the Nobel Prize in 1993.
Wow, thank you. And from this presentation, what stuck out to me, I know you were mentioning in the start that failure is very important, but
Mm-hmm.
going through your presentation, I saw that what was equally as important is that in the face of failure, you kept going. And
Yeah,
I think
right.
to me, it's not luck is very important, but I see that
Mm-hmm.
you kept going in the face of failure. And if you didn't do that, then um, the fate of the world would have been very different.
Well, no, somebody else would have found it. So it was just, but this is the key to failure. When you fail, you have to do a post-mortem to find out why you failed. And this is why flying, for instance, is a very, very safe thing to do, because whenever an airplane crashes, the industry goes to tremendous lengths to find out what went wrong because they don't want it to happen all the time. Flying is makes a lot of money for them. And so this is the key. When you fail, you do a post-mortem, you find out why you failed, and then don't fail again. And the importance of luck was that so we did the experiments, but the final result could have been something very trivial that we'd not even thought about, but it was big. And that was the piece of luck because that led to this huge discovery about nature and what nature was doing and led to the Nobel Prize.
Yes, that's very interesting. Thank you. Well, thank you, Dr. Roberts, for joining us on the Compendium of Knowledge and inspiring us all. It was a privilege to have you here with us.
Okay, well, it's been a pleasure, and I wish all the students there the very best going forward. Lots of luck, and be sure to do a post-mortem on any failures you have.
Of course. Thank
Failure is a good thing. Don't let your parents ever say failure is not good. That's when you learn.